Assalamu alaikum dear students, it is nice to see you again. Thank you so much for following your own lessons. As like in previous videos, we are going to follow these objectives. In this video you will be able, in order to understand cell cycle, M phase, mitosis, significance of mitosis and also meiosis, and significance of meiosis. So let's talk about it in more details. Before starting your lesson, let's have a question. Question is like this, that are you aware that all organisms, even the largest, started their life from a single cell? In order to answer this question, you should be well familiar with two different types of cells that you can find within your body or any kind of living beings which are composed of. So totally we possess two kind of cells. One that is called diploid cell, that possess two sets of chromosomes, and next is called haploid cells that possess only one set of chromosomes. In this picture here I'm pointing, you're seeing two haploid cells which possess each one set of chromosomes. This one is called a sperm cell, then this one is called ovule. So in this case, after fusing or fertilization, you will have another important cell that is called zygote. Zygote is a diploid cell that in furthermore divisions you will have the embryo, or you will have your own self. It means if you see yourself, you can find two important type of cells. One that is forming your own body, any kind of structures and organs that you can see inside your body and outside of your body. So they are composed of diploid cells, except of sperms and egg cells, that they are differentiated from diploid cell and they are called haploid cells. So by fusion of these two single cells, you will have the entire human. Let's talk about it in more details, how we are going to evolve from one cell into multicellular organisms and to be a human. In cell cycle, we will study that the cell cycle is the series of events that take place in a cell leading to its replication. Mainly this circle is composed of two important phases. The first is called interphase and the second is called M phase or mitotic phase, and we can abbreviate it into EPMAT. EPMAT means interphase, prophase, metaphase, anaphase, and the final one, telophase. So different type of metabiological activities are going to be happen inside the cell cycle in order to produce multicellular organisms. So for this to occur, you should be well familiar with the sequence and steps of cell cycle. So the first thing that we see here, the G1S and G2, it is called interphase. So the second step, as you see here, which are divided into subsequences, they are called M phase or mitosis phase. The cell division starts from karyokinesis. Karyon means nucleus and kinesis means division. It means when a cell wants to divide it, it starts its division from the nucleus. Then it will followed by the cytokinesis, which is referring for cytoplasm division. Let's talk about it in more details. So dear students, now pay attention. This is a diagram that ensures you which kind of metabiological activities are going to be happen inside the cell cycle. So G1S, G2 or M phases, these are the important phases that are going to be happen inside the cell cycle. Let's start with a G1. And G1 cell cycle period, mainly the cell are associated with production or synthesis of important proteins, enzymes, and also new organs or organelles are going to be produced in G1 period of phase. So in S phase or synthesis phase, the only thing an important or major effect of this S phase is this, that in this period the DNA is gonna replicate. Their genetic materials are being doubled and allowing there for more DNA for a cell to split into extra daughter cells. In G2, you will be familiar that all process of the cell cycle are going to be checked once again in order to prevent the cell cycle if it has any kind of damages. So G2 is mainly known as checkpoint of the cell cycle. Before entering the M phase and cell checkups any kind of damages, and it reduce it. So when the cell enters the M phase, the M phase are divided into some sub-steps that you should be familiar with properly. It starts from prophase. Mainly in prophase, the chromosome or the DNA molecule is going to be condensed, that which is visible under the light microscope. And metaphase, in metaphase, it is obviously clear from its name, the condensed chromosomes are going to be aligned at the equator of the cell in order to produce a line of chromosomes. Then in anaphase, the centrioles are which have been separated and got its place into two pools of the cell. Here they will produce spindle fibers with a touch with a centromere of the chromosome in order to split two chromatid sisters from one chromosome, and then these chromatid sisters are gonna move toward two pools of the cell. And telophase, which is known as the final stage of the cell division, here cytokinesis happens. It means the nucleus and its membranes, different type of organelles, are reformed. So only remaining thing is the cytoplasm. In this case, in cytokinesis, cytoplasm is also split and it is divided into two different cells. Thank you so much, dear students. See you on cell cycle part 2, mitosis.